Good afternoon and welcome to our Cellular Therapy Series, Part 1, How Engineered Cellular Therapies Are Reshaping Medicine. We are pleased to present you this series in partnership with Bristol Myers Squibb. Stay tuned for Part 2 in September. I am Dale Willard, Director of Member Services for Life Science Washington. For those who are unfamiliar with Life Science Washington, we connect our over 500 member organizations to each other and to funding, talent, partners, and essential business services. We elevate our industry by promoting policies that strengthen the environment for groundbreaking research and product development that improves and saves lives. I'd like to extend a special thanks to our sponsors, Bristol Myers Squibb and Seattle Children's Foundation. As we will hear today, both organizations jumped into the cell therapy research before it was sexy and are now leaders in the field because of it. We are honored to have their support, and this event is free because of it. A couple of housekeeping notes. One, for the audience, please double check you are muted and make sure you stay that way. The goal will be at the end to do a few audience questions. Please, you can submit your questions through the chat during the event. Allow me to introduce today's great cast of speakers. It is an honor to have them with us today. Let's start off with Tina Albertson. She is Chief Medical Officer and Head of Development at Lyell Immunopharma. She held previous positions as Vice President of Juno Therapeutics and Pediatric Oncologist at Seattle Children's. We also have Alicia Collins, Executive Director of Cell Therapy Global Patient Operations at Bristol Myers Squibb. She was previously Director of Cell Therapy Supply Chain Program Management at Juno Therapeutics. We also have Heidi Gan, Chief Technology Officer at Sonoma Bio. She was previously Senior Vice President of Operations at Dendreon. We have Mike Jensen, Vice President and Chief Therapeutics Officer at Seattle Children's. He is a co-founder of Juno Therapeutics and Umoja Biopharma. Last, but most importantly, I want to introduce you to our moderator, Dr. Charlotte Schubert. She is a reporter at GeekWire covering life sciences, health, and biotech. She earned her PhD at the Fred Hutch. She has also worked at an epigenomics lab at the Fred Hutch, a microbiology lab at the University of Washington, and quite notably in Mike Jensen's group at Seattle Children's. She is a former editor and Washington, D.C. correspondent at Nature Medicine and recently worked as a science editor for a cellular immunotherapy group at Seattle Children's Research Institute. Charlotte, are you there and ready to go?
Yes.
Okay, I'm going to stop you. All yours.
Okay. All right. With this panel discussion, we're going to go back a bit into the history of cell therapy in Seattle and then move into the present. But first, let me provide the broad outlines. This is the journalist overview, and we'll have our experts flesh all this out in the next hour. Cell therapy is complex. It requires specialized knowledge to develop, a lot of different components to manufacture. It comes from cells of the body. It's not easy to develop or to make. And that's one reason we have an ecosystem of people in Seattle who know how to do it, and that they've grown, and that ecosystem has grown and stayed here because of the complexity of making these happen, and it needs a lot of expertise. Here, for example, is the outlines of CAR T-cell therapy on the left. T cells are isolated from the patient. They're engineered to express what is called a CAR. That enables the cells to recognize the tumor. The cells are grown outside the body, and then they're infused back in where they home to tumors. I put Juno Therapeutics in the middle of this early cell therapy ecosystem on the right because it was central to the development of CAR T-cell therapy that came out of Fred Hutch and Seattle Children's, and it was not born in a vacuum. Our early biotech companies, and these are only some of them, provided talent for Juno. One of them was Dendreon, which developed the first for prostate cancer. Many of the people trained at Dendreon and these other early biotechs went on to work at Juno, including Juno's CEO, Hans Bishop, and some of our panelists. Juno's pipeline was ultimately acquired by Bristol Myers Squibb after Juno was first acquired by Celgene. So this is a little history here. BMS brought Juno's lead CAR T-cell therapy to the finish line, gaining FDA approval in 2021. That therapy is called Breyanzi. Another CAR T-cell therapy, Abecma, was also approved that year. That was developed by Bristol Myers Squibb in collaboration with Bluebird, which is based in Boston, but also has long had operations here in Seattle. Meanwhile, Breyanzi and Abecma have joined a growing group of CAR T-cell therapies that have been approved by the FDA here on the left, made by other biopharma companies. There's 6 approved therapies now for various forms of blood cancer. Some of the data supporting approval of these treatments has been spectacular. The therapy that became Breyanzi was tested in one trial at Seattle Children's. More than 90% of patients showed an initial remission, and these were patients with few to no options left. But cell therapies are still only approved primarily for patients who have failed other treatments and only in blood cancers. Ultimately, for most of these therapies, less than about half have a durable remission that lasts years. There's a lot left to do. And there's a host of new companies that have grown up to do it. Many of them here in Seattle or that have operations here on the right. Researchers are testing therapies in clinical trials to expand the patient pool for blood cancers. Some like Sonoma and GentiBio are testing cell therapies against autoimmune inflammatory conditions. And many are aiming for the next frontier, which is solid tumors like breast or pancreatic cancer. And almost everyone is thinking about how to make these therapies less expensive, easier to make, and more accessible. All of this is taking place in our region, which has seen an acceleration of biotech activity over the last few years, an explosion of new cell and gene therapy companies, only some of which are listed here. And with that, let me turn to our panelists. If you could each spend about 2 minutes talking about what you do and your background. And I thought maybe we could just start alphabetically with Tina, Alicia, Heidi, and then Mike.
Great, thank you very much for having me. I'm Tina Albertson. I'm now CMO and Head of Development at Lyell Immunopharma. I was previously at Juno, Celgene, BMS, so many of you on the call I know very well. Welcome. I have since moved on to Lyell Immunopharma, where we are focusing on overcoming the barriers of cell therapy in solid tumors. And we focus primarily on cell health and counter-exhaustion technologies, trying to get these cells, both during manufacturing, to have more stem-like qualities, as well as once they've been infused and meet the solid tumor microenvironment, to stay active longer, since we've learned a lot in heme malignancies about needing functional cells for a long period of time to have durable remissions. That's really now what my role at Lyell is leading the clinical development of next generation CAR, TCR, and TIL products using these technologies, really focusing on the cell. And then once we get the cells right, hopefully broadening targets. I'm a pediatric oncologist by training, so I am part of the ecosystem that is Seattle. I trained clinically at Seattle Children's, got my PhD at UW, did transplant work at the Hutch, and started my industry career at Seattle Genetics, where then I moved to Juno, and now I'm at Lyell. And as you'll hear today, we've learned a lot about cell therapy, and both the academic and industry ecosystem here in Seattle has continued to grow, and many of us continue to work together and pull people and technology from Seattle institutions. So I'm really glad to be here. Thank you.
Hi, everyone. My name is Alicia Collins, and I'm currently, as was mentioned, the head of our global patient operations for cell therapy. And we're really managing and orchestrating the patient cell supply chain from end to end. focusing on our apheresis operations and our scheduling, our patient scheduling teams with some other support teams in there for systems and processes. I joined, I guess it was Juno / Celgene in 2018. It was still kind of switching over. And that was my first time in the Seattle area working. And I have to say, I was highly attracted to the area. I think that's actually what attracted, if you weren't already here, was attracting folks to be able to work on really innovative, cutting edge technologies and really being able to add value and feel like you're making a difference in a little bit of a smaller group, and feels very much like a family. So started out in supply chain and cell therapy, and yes, patient operations has a feel of supply chain to it, but did a lot of the raw material planning and capacity planning for the manufacturing sites. And prior to that, I was down in the Bay Area at Genentech and Roche. So thank you for having me here today. Looking forward to the discussion.
Good afternoon. I am Heidi Gan. I am the Chief Technical Officer for Sonoma Bio Therapeutics. I've spent the last 30 years in biotech, primarily in Seattle. I am a Seattle native, was born here. My original training is at the University of Washington, where I have an undergraduate degree in cell and molecular biology, and did a graduate degree in bioengineering. And later got an MBA there also. So I have 3 degrees from the University of Washington. Go Huskies. I then started my career when it was really in Seattle. We're just trying to build the biotech infrastructure, was at Immunex back in the early 90s. And so I spent 10 years there, and I actually started in the laboratory, but worked my way over into operations. So I spent most of my career in the operations of biotech. Last position I held there was chief of staff for global operations for the SVP of operations at that time. I then moved over to Dendreon, where I was the project lead for Provenge, the first active cellular immunotherapy that was approved in 2010. And it came through a Seattle company. So I'd say that was also a kind of a ground landmark opportunity for Seattle in the growth of biotech. I was the senior vice president of operations there, responsible for building out all of our global facilities, patient operations, and all the systems to support that. Post of that, actually thought about taking a little bit of time off, and I did, and got recruited back into the business at the time. As most people know, in the early 2010s, cell therapy was taking off. I was recruited to Sodio, where I was a global COO. As a company out of Prague in Europe. I moved to Boston for a few years, a facility in China, a facility in Prague, and actually worked with all of our trials cross-functionally in the United States and across Asia and Europe. During the second half of that decade, I worked with a lot of investors. As most people know, the funding was really growing and going into personalized therapies. And so did due diligence for about a dozen different VCs and investors across the world, going into personalized medicine, but primarily cell therapy. During that time, I also co-founded a company called Venetti. It is a company that is based in the cloud and does the full cell coordination from order to delivery of products that are of cell therapy nature or of a personalized nature. And then post of that, joined Sonoma. Well, actually, I was a CEO at Zio Farm, came down from the board to do that position for about 9 months. It's a publicly traded TCRT company using Sleeping Beauty, non-viral approach to it, and recently joined Sonoma back about 4 or 5 months ago. I additionally also sit on the boards of Vericel, which has the first 2 cell therapies that were approved a long time ago, back in the 90s. And those are actually Macy, Carticel, as it used to be, and Epicel. And I also sit on the board of Obsidian, which is a private company in Cambridge doing genetically modified TILs. And at Sonoma, we're using using, I would say, all of the knowledge and experience, both in Seattle and across the world, to do TCR-T and CAR-T products for regulatory T cells in autoimmune. So we're going into some new indications and using what I'll call next generation, third generation, fourth generation approaches to it. So looking to be innovative and continue the Seattle tradition.
Hey, good afternoon. This is Michael Jensen. I've been in the CAR-T developmental space for the last 25 years. My journey started getting training as a pediatric oncologist here in Seattle, and I had the good fortune of postdocing in Phil Greenberg's lab, who at that time with Dr. Stan Riddell were really pioneering the adoptive transfer of antigen-specific T cells into humans. I got my first academic posting at City of Hope, and in 1997 set up a CAR T cell research program. And during that time, we launched human trials for lymphoma, glioblastoma, and pediatric neuroblastoma. I had a great opportunity to come back to Seattle in 2010, and with Stan Riddell, we began to set up Seattle's CAR T cell programs at the Fred Hutch and Seattle Children's. More recently, Seattle Children's has allowed me to take all the disparate infrastructure you need to develop and deploy in clinical trials CAR T cell products, and been able to take those assets and vertically integrate them into an end-to-end biotech-like endeavor housed within the academic non-for-profit venue of Seattle Children's. And during that time, we've had 14 INDs approved in first-in-human therapies in pediatric leukemias, lymphomas, solid tumors, and brain tumors, and have been able to expand into a North America deployment where we make cells in Seattle and we ship them back to children's hospitals across the United States and Canada to participate in our phase 1 and phase 2 clinical programs. It's great to be here with you today.
Great, thanks everyone. So we'll start getting into our first questions here. All of you have very deep connections to the region, and that's one reason you're here on the panel. So you can tell us a little bit about how Seattle's talent pool helped build what we have today. How did it help build companies like Juno Therapeutics and our raft of cell therapy companies? And I'd like to point out that some of our, a lot of our region's workers came from Dendreon. Some came from an even earlier cell therapy company, Targeted Genetics, which was founded in 1989. And some came out of ICOS and Immunex, our very early flagship cell therapy companies, and even Cgen, which was founded as Seattle Genetics in 1997. I'll start by asking Heidi, and then expand to our other panelists. Since Heidi was at Dendreon, tell us a bit about Dendreon's influence, as well as the other early Seattle companies, and then we'll move on to everyone else.
Sure. So I think if I take the roots of Dendreon, it comes from a couple co-founders that came from Immunex. So if you go back in time, Immunex was one of those flagship biotech companies you discussed, Charlotte, that really enabled a large talent pool to be brought to Seattle. And it was very science-based. It was one of the largest patent portfolios for molecules that we all kind of use every day today. IL-15, IL-1 beta, IL-4 receptor, Flaherty ligand, GM-CSF. That was only a partial part of the portfolio. It was really based in, I think, some deep molecular science. And what came from that, I think, was the ability is post of Amgen, such an attractive company. Amgen purchased it and actually had home here in Seattle for a while, but really enabled a lot of companies to spur off from that. And, kind of some of the lists we mentioned already before, but just to kind of reemphasize, as you have Dendreon, of course, I said similar co-founders, but ICOS actually had a similar co-founder from Immunex and Dendreon, that'd be Chris Honey. Targeted Genetics, as you mentioned, was a spinoff out of Immunex. It was directly came from the laboratories there, Excite Therapies, Corixa, and of course you mentioned Seattle Genetics. So I think during the 90s, there was just a blossoming of opportunity for these companies. And what's interesting about Seattle is, what's great about it is it's not just, it's deep, deep science. It's not science for science sake. We've created a lot of commercial products that are still sold today on the market very successfully. We've got Enbrel, Leukine, a whole host of them out of Seattle Genetics. And so I think that just created a base of talent and opportunity. And I remember just one quick story is that at Dendreon, when we were doing the FDA inspections for our facilities, and it was the first one, and it was the, PLI, and Tom Finn, who was our reviewer, we kind of felt like we kept getting beat up all week. And we were having all these questions, and we'd all gone through the recombinant protein process before for these sort of inspections, but this was the first of its kind. And he kind of saw us, getting a little worn down and said, just so you know, these products are going to be so important to patients in the future, and you're the first ones doing it. So we are going to ask a lot of questions, and we're going to ask a lot of you. And it's because this is how important it is for the future. And I'll never forget that, because I think that just reflects what has happened in Seattle since then, is you've had, companies like Juno, who's just a huge landmark opportunity there with the CAR T products, Sonoma Bio Therapeutics, which I'm working with now, just all kinds of innovative cell therapies that have come along out of our academic institutions and have seeded other organizations. So I think it really did begin back once upon a time with a talent base, but there was also a lot of cooperation and help with, I think the FDA and others along that really gave us the credibility to create these commercial products. Of course, we keep getting bought out by other companies, but the reality is I think that's just a mark of success. And so I do think that's a little of the story about how it went, and I think how important it is for Seattle and will continue to be so.
Can I add to that, Charlotte?
Yeah, please.
Yeah, I just want to add, I absolutely agree with everything that was said, but one of the key things within Seattle, I think, is the Fred Hutchinson Cancer Research Center and its commitment to transplant some of the earliest cell therapy, with the anti-leukemic effect of allogeneic stem cell transplant. And so I think it's such a great environment here in Seattle, where we have all of the clinical expertise of cell therapy. We have the industry that has grown over the last 3 decades that has the regulatory and industrialization expertise of taking academic products and processes and making them into things that can be commercialized, as well as the tech. And I'll just add, I think it has been a big part of the success of especially cell therapy, and Alicia can probably speak to this more, the technology that's required to take a patient's cells from them, manufacture them, and put them back into the patient in global trials requires technology. And I don't know that I know the exact number of data points from every product we characterize and every patient we treat, but there's probably tens of thousands of data points in cell therapy, which is much bigger than what you do with off-the-shelf therapies. And so the data involved that you have to submit to the FDA, that you have to analyze to try to understand why these cell therapies work and don't work, requires things like Amazon cloud services and the technology of computing big data. And I think Seattle has really pioneered that aspect as well. And those three coming together, the clinical expertise, the industry power, and the technology, I think really have led to huge success that I'm sure will continue here in the Seattle area.
I'd like to move on and talk a little more about our academic institutions, but I also didn't get a chance to hear from our other 2 panelists. If they have anything more to say about how our biotech ecosystem kind of helped build this from the start.
Yeah, I could weigh in a little bit. I think, when you ask yourself, what's the secret sauce about Seattle? Why is the soil so rich? I want to echo Tina's comment that the backbone historically of that was through the robustness of the academic environments, and particularly at the Hutch with their allotransplant and TIL programs and donor lymphocyte infusions, they really were pioneers in a sense where they set up critical infrastructure for translation that is not very often built out because of its expense in the academic venue. So GMP facilities and regulatory affairs know-how. When I came up to Seattle, I very rapidly put together those pieces, and we occupied the facility that was formerly run by Targeted Genetics to have clean rooms to start these programs. And when you think back at Juno as an early pioneering commercialization effort, that really came out of the trials that Stan and I were able to launch. And Bob Nelson famously was on the board of the Hutch and was getting a board report on the activity of these cells in leukemia and lymphoma. And he stood up and said, this has to become a commercial entity to basically harness the potential of this therapy to make it available for patients based on a prescription. And so, you know, that was a famous landmark where Bob put the stake in the ground, and Juno came from that vision.
Yeah, I want to follow up a little bit with that. And just for our audience, Bob Nelson is at Arch Venture Partners, which is a big backer of Juno. Juno Therapeutics emerged from technology developed at Seattle Children's, Fred Hutch, and Memorial Sloan Kettering. And you told us a lot about the background of why Hutch was well positioned to study this area with this background in bone marrow transplantation. But can you tell us a little more about the details? Where did that tech come from that made Juno?
Yeah, I think sometimes it starts with a very few people in academia that become like-minded. So, you know, Stan Riddell is a pioneer in adoptive T-cell therapy. And I was doing very early CAR T-cell trials with first-generation technologies starting in 2002 at City of Hope. And we basically were collaborating remotely. And then I had the chance to come back to Seattle. And I remember rolling into Stan's office with a trunk load of INDs and SOPs in order to have the Hutch be able to really fast start CAR T-cell manufacturing. At the same time, I went after pediatric indications through the Seattle Children's Program. And that was in July of 2010. And I remember almost exactly that interaction of coming back onto the Hutch campus with those materials and Stan and I stacking hands and saying, we got to make this go. And that begat clinical trials and that begat the impetus to launch Juno Therapeutics.
So it all happened. You had your INDs, that's investigational new drug applications, and your SOPs, which is your sort of roadmap for how to do it. And then and it started to come together. And then once you formed the company, there was still a lot of challenges to overcome. And this is for everyone who's been at Juno or is affiliated with it on our panel. I'd like to know a little bit about, you know, what Juno built and what kind of scientific and logistical challenges it had to overcome. So I'll just open this up. Maybe start with Mike and then we can move on.
Yeah, I think, you know, initially, Juno started very much from let's capture the progress and value of what was happening at the Hutch and Children's, and then at a later date brought Memorial Sloan Kettering in. So they actually had 3 shots on goal for a CD19 product, in which the product at each institution had very important different variables, whether the CAR construct had a 28 zeta or 41BB, whether the product was manufactured from purified central memory cells or balanced 4 and 8 cells. So I think, you know, very early on, and I'd love to hear from the folks that were in Juno, they had a little bit of an embarrassment of riches with 3 products that they could look at in terms of real-time data from clinical trials and then make a decision on their favorite to move forward for a commercial product.
Yeah, let me just add to the clinical, and then Alicia can probably speak to kind of the operational challenges Juno had. As Mike mentioned, we had 3 CD19 targeted agents that were from different academic institutions. One was from MSK. That was the one that was JCAR15 and went directly into pivotal trial in adult ALL and met some clinical challenges that I can speak to a little bit in a second. The other two were highly related. They used the same vector. They were CD19 targeting with a 4-1BB co-stim, slightly different manufacturing. One was the one Mike Jensen was very involved or used at Children's. And the other was what Stan Riddell and Cam Turtle were developing at the Hutch. And so we had a lot of data to look at, but we also had a lot of academic collaborations where our ability to ask clinical questions was broadened because they continued their clinical trials while we ramped up and commercialized processes to run our pivotal trials that would lead ultimately to the FDA approval of JCAR17, the one that came out of Mike Jensen's lab at the Hutch. I'll just talk about a couple challenges since that was the direct question, just to give people a sense of what scientific challenges can happen. We took a pediatric ALL process. So
do you mind if I interrupt just for a second? I just want to make sure for our audience who's not that familiar with cell therapy is that CARs go to a target, and one of the first targets you guys looked at was CD19, and that's on B cells. So that homes to the B cell. And then when you talk about co-stimulation, it's another thing you kind of add to soup the cells up. Did I got that right?
Exactly.
Okay, cool.
And the co-stimulation has kind of two flavors, a CD28, which can be more of a quick and a very fast expansion. It's actually usually a little more toxic, at least in B cell malignancies. And the 4-1BB is we call a little bit more of a slow burn. The cells take a little longer to expand and reach remission, but are a little less toxic and tend to last longer in the body. And so we had some options and they had different clinical profiles. But interestingly, even the patient population mattered. So Juno took a pediatric ALL process and tried to put it in adult patients with ALL. And I think we failed 2 out of our first 3 lots because T cells from a 70-year-old lymphoma patient are not the same as a 5-year-old pediatric patient. And so we had to quickly adjust. And this was in an active clinical trial, and had to work quickly with the FDA to modify and take out our selection process on the EGFR tag. And so we didn't have a pure CAR T cell product, which is very different than what was started at Children's. And we actually had a back and forth with Children's, and Mike can probably speak to his side of this, where they eventually stopped selecting, I think, as well, based on the data we did, because the manufacturing became much easier. So it's just an example of where the academic process in one population couldn't be industrialized. And so we had to, on the fly, make some pretty major changes in manufacturing that had effect both to the trial as well as our manufacturing success. And most of you know as well that we had some challenges with JCAR15 in adult ALL, mostly because as we industrialized the process from MSK, we actually made it better and better. As we controlled the process more and more, the cells actually got healthier, and we were likely infusing a more active functional dose of cells than they had been testing at MSK. And so these are just some of the scientific challenges we had to deal with very quickly because patient safety was on the line and patient treatment. And Alicia, I don't know if you want to add anything to that, or Mike.
Yeah, sure. Again, I mentioned I came in 2018, so standing on the shoulders of the folks who had done the work up until then, and there still was a ton to do in 2018. And if you think about the whole process, your starting material is the patient's cells. So how do you get that in a very quick way to the manufacturing site, get it manufactured and back to them, tracking it the whole time and making sure nothing goes wrong is quite the challenge. In terms of manufacturing, it's very labor intensive. The first time I saw the manufacturing site, it's not big tanks. It's little equipment that looks like you're in a lab, and it's interesting to see that in a manufacturing setting. You don't scale up, you scale out. So if you want to do more, you add more people, and those people require space and training. And how do you plan for what capacity you're going to need? Going from 1 or 2 patients to 20 patients a week, 50 patients a week. That's quite the big leap. And so getting everything, thinking about that and how you want to commercialize, And trying to do that at the same time where you're supplying clinical trials and getting ready for commercial is, it takes a lot of people. And if you think about the suppliers that we use for raw materials and vector, very nascent, not a ton of maturity. So you have to help them mature, grow with them, and bring them along for the ride. And the whole time making sure too that your quality systems are in place to do rapid product release. There's not a lot of time here. There's no buffer. We don't make this and put it on a shelf. It's made to order. So, you know, going back to learning as you go, changing things as you go, that was often the case. You work on something, you get it all set up. Shoot, we didn't know about this. Now we got to go back and work on it some more or change it completely. So it does require a lot of coordination. It requires a lot of cooperation. And that's when I was saying in the beginning, it feels like a family. It just really feels like everyone's working towards this common goal. Very complicated, but also everyone's really, you know, on board and really wanting to make it happen. And there are so many logistical things that you have to think about getting set up to execute on that, on what I just said, and be reliable for the patients.
Yeah, when you mentioned
I would just add that what was really eye-opening to many of us was that in contrast to making a small molecule drug that once you manufacture it and purify it and formulate it, it is the drug substance, and it has, it's basically fixed in terms of its therapeutic index. With cell therapies, what we learned really quickly, and we're still learning, is that the therapeutic profile of the drug, the cell product, is hugely influenced by the way you manufacture it. Even though products in the end could look highly similar in terms of cell subsets and CAR expression, that even small changes in how you manufacture them, what stimulation bead you use, what cytokine mixture you use, can have very, very significant impact on the therapeutic index of that product. And that I don't think was fully appreciated before we got into larger scale CAR T manufacturing programs.
One thing you mentioned, just as part of the complexity, is something called the vector, which is the thing that delivers the CAR into the cell. And that's just one example of all the different moving parts. I don't know who the right person is to ask about this, but I was wondering, can you talk a little bit about the regulatory path forward with the FDA, how you worked with them to help them understand the product and work through, you know, what you needed to do to get the product moving towards approval?
I can speak to that from the Juno standpoint. And as I mentioned before, manufacturing changes and adjustments were part of our life. It was probably the biggest hurdle with the FDA as well. And we had to teach them as we went why and how it mattered. And eventually we actually got them to be quite flexible on what they would allow. As you may or may not know, other folks on this call, if you work, as others have mentioned, in small molecule or even biologics, you have large lots of drug that are all the same. And so once you show them that you have comparability between lots, you're good to go. With cell therapy, you're showing groups of patients or groups of manufacturing lots are comparable with wide variability between patients. And we had to get them comfortable with that uncertainty. And they eventually did, because we had to show them the outcomes of patients were similar. So we made, as an example, for JCAR17, which became Breyanzi, we had 4 versions of the drug that went into our BLA. 3 of them, they eventually allowed us to call the same thing. We had to throw out, I think, those first 3 patients that I mentioned that had the pediatric manufacturing process. But the other 3 had fairly significant manufacturing changes. And probably the biggest hurdle we had to do was show them that although it changed the product some, it didn't change the clinical outcomes. And so this was part of the clinical trial where we had to modify and actually show clinical comparability in addition to manufacturing comparability. The other thing that I think we really pushed the envelope on in clinical development was patient population. Usually the FDA requires you be very narrow in your patient population, but we knew that would limit how many patients could get these life-changing therapies. And so our trial, we actually designed to have almost all adult lymphomas included in the trial design. And we eventually got all those patients into the label once we showed the FDA the benefit in those patients. And so I think in cell therapy, we were given the ability to be more bold. And I think we need to continue to do that just because of how much benefit can come from this kind of therapy. We don't often see curative therapies in oncology.
So this question, sort of to bring out any stories, if you have any, about Juno, which is, or did you ever have a moment where you thought, where you were just really felt frustrated, like this isn't going to happen? You just felt stymied, and what did you do to overcome it? I don't know if you did have that moment, maybe you didn't, but if anyone has any stories to share about that.
I can have a quick one here. Of course, yes. It's so new, right? It's not very many people have done it, and this is maybe a little cliche, but you don't know what you don't know, and not everything's under your control. So again, on the operations and logistics side, I think certainly there are setbacks, and certainly, you think you're getting a certain supply of something and you're not getting it, or the process is more complicated and it's not processing, or it's not moving forward as you were expecting. So you go back to the drawing board, and there certainly are disappointments along the way, but clearly there's a light at the end of the tunnel, and you eventually get there. Everybody kind of takes a step back and says, okay, let's just have this moment for a second to be disappointed, but then tomorrow let's get back to work and let's get everything moving for the patients. And again, it's really motivating to have that, as to know that you're helping these patients, and so people don't give up. And you know that it's going to happen. I think that's part of cell therapy, is you don't know everything, and you know these things are going to happen, but that's why there's a lot of smart people working here, so that we can solve these problems and keep creating great products for the patients.
I want to move on and find out what BMS did next, but if anyone has any other stories or thoughts they'd like to weigh in about Juno and its construction before I move on.
I could share some funny stories of what you do with a group of cranky academic founders once the company gets going, but that's for another time, Charlotte.
Okay, we'll be sure to make another time. All right, this question is for Alicia. We're moving on because ultimately Juno's pipeline was acquired by BMS, which has maintained its operations here in Seattle and grown. So tell us a little bit. In addition, BMS also developed Abecma in partnership with Bluebird, which is now 270. It's 270 Bio. And then, so my question for you, Alicia, is what infrastructure did BMS have to build or tap into to go to the next step and take Breyanzi and Abecma to market? Maybe in a couple minutes, tell us what that happened.
Sure, and I'm sure Heidi can comment on some of this too from her previous experience. And it's a lot of the things that I've already mentioned, but it's also, you have to be very forward-looking. This does not happen quickly, right? So currently BMS is building 2 new sites, and they won't see patient for years, right? Because it's such a long lead time. And as I mentioned, you don't just go buy bigger tanks, because that doesn't help you. It's individualized medicine. So you're stuck with scaling out and not scaling up. And so really it comes down to planning, investment. It's not inexpensive to commercialize cell therapies, and you have to be pretty committed to it to make sure that it happens, and it happens in the right way. You really have to hire a lot of people, and so how is that going to work for you? And I can tell you, as I mentioned, you're not going to think of everything. So one of the interesting problems we had was we had this really great large manufacturing site, and there were so many people that needed to be in there to make the product that the people flows were clogging up the locker rooms. And so we actually had to do construction on the locker rooms to make those bigger so that we could manufacture product and accommodate all the shift changes and all the operators that had to come in and out. So a lot of being able to take it from early phase clinical trial and into commercial product is getting those processes in place that support commercial operations. There's not as much flexibility, right? And then again, it's so critical. It's so critical to plan ahead, much more so than in the traditional modalities of biologics and small molecules. Really have to have a vision, and maybe it's wrong, but you have to have something and you need to plan towards it to get all those things in place with those super long lead times. So, you know, when BMS did buy Celgene Juno, it really is providing, right, what is needed to take this to the next level, and really having that strong investment in cell therapy from BMS is excellent.
All right, thank you.
I think I'll just add to that to say is that I agree completely. And one thing I think we talked a little bit about earlier on was the high-tech aspect of this. So these are not just physical products, they're digital products. In fact, the physical part of it is almost easier than the digital part of it. And that is that you have everything happening within a clinical site, the ability to order it, then to pre-clear it through all the insurance, then to be able to schedule, to do the transport of cells, have a manufacturing slot available, do the shift scheduling for all of the resources throughout. So what we're talking about here is as much a digital product as it is a physical product. And I think we're now, you know, back when we were doing Provenge originally, we built a system internally and put it on-prem, which really would not work. And that's on-premise, meaning that you have your own servers there and you have your own coders. You know, I say all my gray hair comes from that, not from anything having to do with the actual physical product. It's something that is not in the wheelhouse of biotech. And so it's something that all of us have had to learn to kind of integrate that high-tech aspect with the cellular product aspect. And it's really how you make sure you have good customer service. It's how you make sure claims are paid for in a commercial world. And when you have a high volume product, so for instance, Provenge, up to 25,000 doses a year go through these facilities on shift schedules of 24/7, that requires a lot of digital information and coordination to make it happen and make sure that patients get their product and that eventually claims get paid. And so I would say that's the other piece. It's been a big learning aspect for the whole cell therapy industry. And I think we'll continue to be able to take advantage of things like AI and predictive learning, which we're, you know, using, but only in a nation fashion. And I think we can use that a bit more as we go forward, because it will really help. It will help us get to more patients and get greater, broader access, which is what we need, actually, especially with cell therapy.
Yeah, and I think I'll add in terms of kind of cell therapy in the nascent CAR T environment. It went from overnight, basically from an academic manufacturing, which is typically GMPs with tiny little rooms with one hood, one centrifuge, and one incubator, to needing to be scaled out. And secondly, the manufacturing really rapidly had to change from, we made CAR T cells with whatever tissue culture ware, maybe developed by blood banks for platelets or centrifuges, et cetera. There wasn't a physical plant notion of how to do scaled manufacturing, and there weren't the kind of the basic tools for how to do closed system tissue culture at scale either. And, you know, to Juno's credit, they built the JUMP facility, which really had a fairly forward-looking way of designing ballroom suites for manipulations and then expansion adjacencies. And they very rapidly got serious about kind of bioreactor use that maybe wasn't initially designed for purpose, but thankfully, as time has gone on, there's been more interest in developing the hardware and the tissue culture software for scaled manufacturing. And I think, the field is still way behind the curve in terms of how do we scale manufacturing to the numbers of patients that could benefit per year from these products. We're really behind the curve.
Okay, we have a few more minutes before I turn it over to audience Q&A. So I want to ask everyone the same question, and they can maybe answer it in a minute or two if possible. We talked a lot about, how tech also is involved in this. We have a lot of data you need to, evaluate in these, with these technologies. I'd like everyone to go through the panelists, perhaps again in alphabetical order. How does the cell therapy ecosystem helping you today in the endeavors that you have going on at your company? You can maybe highlight, or your academic institution, highlight one or two key areas you're working on and how the ecosystem kind of helps support it. If we could kind of make it what's called a lightning round, just kind of give us the top line.
Great, thank you. I'll start. So I'm at Lyell Therapeutics or Immunopharma, and as I mentioned before, we're really trying to get into solid tumors. And upon success, compared to CD19, where there's maybe tens of thousands of patients that can be treated, solid tumors, we're in the range of hundreds of thousands of patients eventually. And so scaling becomes even more important. And so I would say the infrastructure, we're working with AWS to start really going that next level to the cloud for both patient management, product management. We also have built our own manufacturing up in Bothell, close to JUMP. We have a lot of people that helped build JUMP, that helped build Life. And so we're completely paperless. It's a lot of automation will be going in, especially to our TIL manufacturing. And so we're using a lot of technology, kind of to these last comments that people were making, to really help us scale in the future. And of course, that takes a lot of investment. And so in terms of forward thinking, we're really trying to invest in technologies that can improve manufacturing times so that we can increase our capacity, because it really is, upon clinical success, how do we get these products to the most patients possible so that as many patients can benefit. So we're really paying a lot of attention to the technology side of it. I think it will help us in the long run as we implement some of those things in the clinical development to try out different ways of doing it, so that we can, by the time we get a product onto the market, understand what's going to be required for that scale of cell therapy manufacturing and treatment.
I will look at it from a talent perspective. I think, you think of certain hubs in the country, I'll say San Francisco, Boston. It feels like Seattle is not just this, there's one or two companies up there now. It feels like it's becoming a hub, and it feels very attractive for incoming talent, right? And I don't think it's a hard sell anymore. And I know for me, I don't want to leave. I love it here. This is the best move I've ever made. And so I feel like other people are seeing that, and it is on the map, right? It is the heart of cell therapy. And it really, I think that goes a long way to continue to attract talent to the area, as well as, building the talent within the area.
So for Sonoma, we have both a site here in Seattle and a site in South San Francisco, and we're going to be growing rapidly our Seattle site over the next year and a half with the intention of doing our own manufacturing. And a bit of both what Tina and Alicia said is we're lessons learned from, I've built over a dozen of these facilities across of the world. And you know what, it's an iterative process of learning. And we're not quite still to the place where we really know what these need to be, but we know that we have to harness the technology that's available. And that's both from a manufacturing and equipment flexibility standpoint. We need to be flexible facilities, but also we need to really have that attention to the digital aspects and really what can we do in terms of predictive learning to really look at, like, the disease indications that we're looking at are autoimmune. So if you think that oncology of solid tumors is large, autoimmune is an overwhelming number. And we're very excited by that, but we also know we have a lot to deliver. So for us, really being able to grow within Seattle and using the talent in the Seattle area is going to be very important for us. And we're also looking to, we've used viral vectors so far, but we've got next generation opportunities coming along. We really see a lot of that developing also and being brought into the Seattle area. So it's a combination of all of that we're expecting to put into our facility that, we plan to have open in the next 1.5 to 2 years. So a lot of growth will happen within Sonoma, and we're very excited about, being highly innovative in areas that nobody's taken cell therapies to yet. And we'll be one of the first with a very targeted product.
Can you give us some of your thoughts, Mike, and then we'll move on to the Q&A? I think you need to unmute.
Just an example of some of the synergies that occur when in the same city you have robust academic programs and biotech that are co-localized in a fairly tight geography in the Lake Union area, as an example. But, for instance, we just built out a 100,000 square foot GMP facility, and the jump ballrooms were really, really instructive for us on how to scale up in the number of products. I think on the flip side, we get young folks out of their PhD programs and their master's programs and their bachelor's programs, and we train them up in a very biotech-like environment in terms of R&D, manufacturing, QA. And I know that my colleagues on this phone and on the call have found some very good employee recruitments coming from the young people that we train in that academic environment. So I think there's symbiosis there that's really important.
Great. I'm going to pass this back to Dale so we can at least get a few Q&As in.
Well, thanks. We got a minute for a question or two. I got one from Kelly Carlton, who asks, for autologous cell therapy, is there a one-size-fits-all manufacturing approach, or do the growth factors and other manufacturing processes need to be customized for each patient? Anybody want to take that one?
Well, you're going to standardize to a certain extent, because what you have to show is a reproducible process that can fit into a statistical set of bars and be able to treat a broad population. However, I'll say broader speaking is that a little of the technology we were just talking about, in academic institutions, both within Seattle and outside of Seattle, there are some great new technologies that are boosting our opportunity to expand cells, to differentiate cells. And so I would say is that when we say next generation, we're talking about taking some of those technologies, putting them into the GMP space and commercializing them. So I would say, yes, the process needs to be somewhat industrial and standard, but we're in the process of looking at how do we make this process better, more optimized with lower cost of goods, and has the ability to use a lot of this digital information we're talking about can help us too. It's going to give us feedback, especially as we treat more patients, to understand really how these different technologies that can sometimes boost different parts of our cell process can give back to us.
I'll just add, I don't think there's a one size fits all yet. As I mentioned earlier, a pediatric process optimized for the pediatrics didn't work in adults. And so you either have to figure out the lowest common denominator, at least for T cells, and optimize a process that will help grow the cells in the worst shape, or you have to tailor to different patient populations. I don't think it's an individual patient necessarily, but often different types of cancers or different patient populations, or certainly different types of cells, T cells versus NK cells versus Tregs, that kind of thing, will need different processes.
Mike, did you want to add something?
Oh, no, I think those comments are right on. I think the way we're manufacturing in 2022, it already looks like a 8-track tape sitting in some old AMC car. They're just the whole industry of automated cell manufacturing processes is just lagged behind because until recently there wasn't an investment thesis on building out those kind of next generation hardware devices to facilitate scaled output. So I really agree. And I think also, we talk about CAR T as if T cells are one thing, and you get into the immunology of T cells, their subsets and their functions. There are hundreds of different types of cells, and we're just scratching at the surface in terms of understanding which cells need to be adapted for what use, whether it's cancer, autoimmunity, or other applications.
I think that's about as much time as we have. Charlotte, do you want to add a few final remarks? Or
I just want to say, I just want to say thanks to all the panelists for a really great discussion.
Yeah, thank you, everybody.
Thanks for having us.
Yeah, our pleasure.
Thank you.
Thank you.
So with that, I want to thank Charlotte and all the panelists. It's been a privilege working and learning from everyone. First, I want to remind everyone that this event was created and produced by Life Science Washington with Bristol Myers Squibb. Second, this event was made free by the generous support of Bristol Myers Squibb and Seattle Children's Foundation. I want to, if everyone can mark their calendars for part 2 of this cellular therapy series, answering the question, what does the next generation of cellular therapies look like? That'll be in September. In addition, we hope to hear perspectives from cell therapy patients and survivors, which will make it very interesting. Life Science Washington has several other upcoming events. Next week, we'll have Device Connect. In 2 weeks, we will have World IP Day. And in June, we'll have an in-person event in Spokane, Washington for the East-West Summit. Go to Life Science Washington homepage and click on events, and you can find more information and register. And with that, I would like to say thank you, everyone, and may the 4th be with you today. Bye-bye.